Thank you, Dr. As we all have already listened the presentation of Dr. Umar, he has, although I joined late, but he has discussed many important things about Alzheimer's disease. Uh, they have used acoustic data to group different Alzheimer patients. They have compared with the control data also. Uh, and then we want to move forward in diagnosis of Alzheimer's as well as all other diseases that are multifactorial. So we cannot ignore the importance of genetics because genetics is the field in which we discuss mostly the basic code that is present in the nuclear genome and whole genome of the individuals. Uh, So when we have acoustic data, and in addition to that, we have genetic data because we know that the DNA is containing the code, and this code is then transcribed in the form of messenger RNAs in the case of protein coding genes. And then this RNA is sent to the cytosol where it is translated in the form of different proteins. And when there are some mutations in those genes, so those mutations are also translated in the proteins, and there are some amino acid changes that then affect the whole structure of the proteins. So if we combine this data, so if you look in the past, so it took more than 15 years, 15 to 20 years to sequence the whole genome. But with the advent of new technologies, which help in gathering the sequence data in a very fast track, so the 20 years period was reduced to only few hours. And nowadays, we can get the genomic data, even whole genome sequence, at a very low cost. So in few years ago, it was very expensive, but nowadays we can sequence whole genome in $400 to $600. And those whole genome data, when we have many patients and we whole genome sequence all those patients, and then we use artificial intelligence to work on those nucleotide sequences. So this artificial intelligence idea is also very old, but there were no computation resources. So nowadays we have very good computation resources, and now we can analyze the data with good computers, with good computation facility. So when we will be getting the whole genome data, then we have different variations, population specific variations, and other variations that are protein coding. So we can combine this acoustic data with those sequence data, and then we can move forward, and we will be able to work out about the disease risk prediction. So because when an individual is already having symptoms, then it is very difficult to treat. But if you have genome data and you use that genome data to identify the individuals who are at risk of having the disease. So when you very early identify those risks, then you can manage the disease in a better way. And nowadays, even if you look at the literature, recent literature, there are many publications which are reporting different variations that have been identified because of the genome data, and combining this with the AI. And then some babies, those who are affected with many different diseases, very severe diseases, they are even securing clinicians are securing their lives, and even based on that data, they are also applying on the other individuals. So similarly, in the case of Alzheimer's disease, we can get the whole exome data, whole genome data, and then we can combine with the phenotypic data. And by analyzing that data, we can reach at some good point where we can pinpoint that this is the variation that is pathogenic, and that pathogenic variation is actually associated with this current condition. So when we will be knowing the uh, condition and its variation that is associated, so genotypic phenotypic correlation, then we will be able to find the solution also. And that solution is also based mostly on the AI, because nowadays we have AI and a human mind cannot think so fast. And when we have these computation resources and AI is combined with that, and we have also phenotypic data. So all these things will combine and they can pinpoint the variations that are associated with the diseases. And based on that, then we can work out how which drugs can be designed to treat these persons. So this is the whole scenario to go forward in this field, not even for the Alzheimer's, but all the multifactorial disorders that are not monogenic. And there are many variations that are behind those diseases that can be treated. And even the individuals who are at risk, they can be identified. And when the at risk population is identified, then it is very easy to even subgroup the people who are having some different symptoms. So we can make the diagnosis correct. Sometimes there are phenotypes, but clinicians are very, they cannot uh, correctly identify that which phenotype and which disease is this. And based on the genomic data, when you have the variations, even you can correct some diagnosis. Clinical diagnosis is not true, but after getting the data and reaching to the pathogenic variation, you can correct the diagnosis also. And based on that, then we can offer the personalized medicine. And nowadays it is practiced in some countries also, although it is not very common in our country. But nowadays, this data is enabling us to move towards the personalized medicine. And we can treat the individuals according to their genome. So I think this is the way forward, and that down Dr. Malia will tell you genetics of the Alzheimer's in her presentation. So I think so 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 what is the time now? How many? 10:20. So if you have any question, you can ask about this. So because this is a panel discussion, so I will not be talking all the time. You people can ask me something if you have any question about what I am talking. So Tumara, have you any question? Okay. So if you have any question, you can ask. So the basic idea is that when you have DNA is translated into transcribed into the form of RNA, and then this RNA is going to the cytosol where it is translated in the form of proteins. So most of the scientists, they know this is the basic dogma, central dogma of the molecular biology also. But when this whole process is going in a normal way, there is no problem. But when mutation is there, then it will be translated into in the form of proteins. And when you have mutations, then phenotypes will appear. There are thousands of genetic disorders. Some are, as I've already said, multifactorial. There are some environmental factors also. There are some genetic factors also. But when you don't have a single gene involved, you cannot reach what is the basic genetic cause. But nowadays, because when if you look in the history, when Watson and Crick first discovered the helical structure, double helical structure of the DNA, then a revolution came into the history of molecular biology and biotechnology and genetic engineering. And even then, till the 1994, when Cariboulos, he invented the PCR. And when he invented the PCR, then another revolution, it was a breakthrough, and another revolution came. And genetic research was going at a very fast pace. But even then, more than 15 years, 15 to 20 years were taken to sequence the whole genome. The whole genome that nowadays we are using in our whole genetic research. That sequence is not of a single individual, but it is a consensus sequence that is being used. So if we did not have that sequence, we could not have reached at this point at which we are today. So when whole genome was sequenced, then molecular biology was going very fast. So in the current century, when different next generation sequencing technologies were invented, so the research was even going more and more fast. So nowadays we can get the sequence of the whole genome. Whole exome is when you are only sequencing the coding part. Coding part means that exonic sequences that are coding the proteins. Because humans are all made up of proteins, and there are thousands of proteins that are involved in different functions. Some are structural proteins, some are functional proteins. So when there are mutations, so mutations are translated in the form of proteins. So when we got new technologies, then we started getting the whole genome sequences. And there are 2 to 4% DNA part that is coding. The rest is non-coding DNA. Non-coding DNA is also very important because that non-coding, there are some there are some RNAs that are involved in different functions, microRNAs, there are ribosomal RNAs, transfer RNAs. So all those are involved in many functions. There are many that are involved in the control of gene expression, because all the tissues, if you have brain tissue, if you have cardiac tissue, you have retinal tissue and many different tissues, hepatic tissue. So they all have different genes expressed. So tissue specific expression, tissue specific gene expression. So if you are, even if you are getting the whole genome sequence, so it is the basic sequence that is present in the nucleus. But when you talk about different tissues, so in the brain, there is a different set of genes that is being expressed. Expression means different proteins. In all cells, there are different functions of different tissues. So they have different genes. Aside from the housekeeping genes that are involved in many common functions in all the cells. So when a tissue specific function is necessary, then you need whole transcriptome sequence also. So transcriptomic studies are also being done, and similarly, metabolomics studies are done. So when you get all these data, so when you will be, for example, if you are working on the brain, just like Alzheimer's disease, you need some cells that are mimicking the brain cells. So for this, scientists are designing different stem cells. The stem cells are those cells that can be reprogrammed to make different types of cells. So if you, because we cannot get the brain cells of a person, that is not ethically correct. So when we get stem cells, those stem cells are engineered to have the normal copy of the gene. And then we can check the effect of that engineering. What is the effect? Either those cells are working properly or they are not working properly. So, therefore, each and every cell tissue of the different organs, they have different sets. And if we combine all those things, so it can be a multi-model thing. You have genomic data, you have transcriptomic data, you also have phenotypic data, you also have biochemical data. So combining all these data, we can reach at a conclusion that can be very much helpful in making the correct diagnosis, and even reaching at a place where we can say that this is the exact nucleotide change that is causing the disease. So if you will be reaching the common nucleotide, that is the main culprit in the gene, then you can work out further by using all these technologies, and you can see that this person is having this phenotype. In this person, this is the problem, and because of this, the individual is having the disease. Then you can move forward. Pharmacogenomic studies, in which different pharmaceutical companies, they are getting these data, and you might have listened that there are many companies that are freely offering the services of genome sequencing and exome sequencing, and then they are using that data to analyze data, and then they will be identifying the genes that are basic cause of that specific phenotype. And then they are analyzing the data further to identify different targets, therapeutic targets. And then they are designing different molecules that can be further used to treat the disease. So this is the whole scenario. And sometimes even if there is some common molecule, you can just offer medicine that can be taken orally. Just as an example, if folic acid is a molecule that is necessary for many processes in the retina. So if a person is having mutation in the gene that is involved in making folic acid, then you can just give that person the folic acid therapy. They can orally take this medicine. And similarly, there are many others. So there was a family, I think in Cuttack, and we treated that family with a chemical, and the patients were having some improvements in their phenotype. Similarly, there are many other examples. So RP65 is a gene, is a retinal-specific gene, in which when the therapy is given, then individuals are having some improvements in their phenotypic. Yes, sir.
Like to very interesting mutation that is actually we are talking about. But I would, if we're talking about the Alzheimer's disease.
Yeah.
So how this one is linking, or how is this mutation process which you are proposing is beneficial for the Alzheimer's disease, Alzheimer's patient, not disease author, Alzheimer's patient. Because I've noticed you said that it can be beneficial for the patient or something early detection.
Yeah.
But for the Alzheimer's patient, how is possible? How is it interpreted? Because it's extremely important, and like nowadays it's very important.
Yeah, stem cell technology. So the stem cell technology is a very important technology. And when stem cells are engineered, so they can be used to treat even those patients who have already got onset of the disease. Although it is important for the early detection and identifying at risk population, but the stem cell therapy, that is also an option that can be used for the patients also. Yeah, and in some cases, even in the retinal disease, there are examples. I have not seen any example about the Alzheimer's disease, but in the case of retinal diseases, I have seen different publication where they are just treating without any corrected copy. They are just using those stem cells to treat the patients that have their eye problems. So similarly, these cells can also be used for the brain patients, just like Alzheimer's patients.
Is it safe? Like for
Yeah, it's safe because
it should not
because the stem cells are derived from the patients. They get the bone marrow of the patients, and they derive the stem cell from those patients also. So because so there is no allergic response. So there is no immunological response. So they are safe. But I have not seen just till now. I have not seen any therapy like this. But it can be done in future. So any other question?
Sure. Excuse me. I just checked you a bit late due to the presentation. My question is, a lot of data you just taking physiological data, maybe speech data, many data, just to give you a system, and system will just for you.
Yeah.
Either the person has the disease is there or not for identification.
Yeah.
As per my knowledge right now, taking from your presentation, Mr. and you as well. At the same time, if you detect the disease of Alzheimer's in between with the Parkinson's and then frontotemporal and dementia.
Yeah.
So we have the imaging technology as well at the end. So we just get the MRI data or CT scan data, and we just try to train those images through deep learning and try to know about what actually the state of the patient is there at the end.
Yeah.
So how your system is different than the imaging data, and it really helps the health practitioners just to identify and just to properly locate the disease inside the brain, or maybe the health.
Yeah, actually, very good question. That how brain data, imaging data is relating to our data, because imaging data is very important because it is a phenotype, but when you are treating just based on the phenotypic data, so there might be some medicines that are not good for different persons. So you might have listened about the non-responsiveness of the medicine. So if a person is non-responsive for a specific medicine, so only imaging data will not be able to cater that patient. And even sometimes when they are continuously taking some diseases, there are some other side effects also. So instead of correcting the real problem, that medicine is creating many more problems also. So, but when we will combine this genomic data as well as biochemical and other data, then we can identify that a particular individual is responder or non-responder for different categories of the medicine. So if you'll be knowing that, then we can treat very good. And the individuals who are respondents, that will be having those therapy, and those who are non-respondents, they will be looked for other options. So this is the beauty of our data, that when we will be combining all this, we can treat the patients very well according to their genome.
Is there any provision of the imaging data to be incorporated with your data for the efficient?
Yes, yes. That is the work that is Grover is doing.
And I want to go and show.
Yeah.
And we are
also showing.
we are also actually going to show you not only the imaging data that we have worked on so far and the results that we have obtained, and also the genetic data that we have worked on. And our next phase is to having an integrated multi-model or multi-modality that Umar actually talked about in his presentation. So in my presentation, or which Umar and I are going to cover, we are going to show you the MRI based diagnostics and AI assisted based, and also the genomic and again AI assisted genomic based data analytics, and how these are actually helping in providing precision in diagnostics modalities. So somehow we are not improving the clinical outcome of the patient or clinical diagnosis of the patient, but also, as Dr. Ajmal said, that we will also be able to pinpoint the responders and not non-responders from our own data for the Alzheimer patient if they are being given some different drugs, especially as it is an old age disease. So we are going to have multiple, I think, phenotypes at that time, because with old age everything is declining and you are susceptible to having multiple phenotypes. So you can also somehow pinpoint those phenotypes from your genomic data and also separate them using MRI and acoustic signaling. So that is what we are planning to do. But the work that we have done so far is MRI based, acoustic based, and genomic based, and model integration is one that is looking forward, and that we are expected to complete in soon.
Yeah, I would like to add some point to your question. Like you were talking about like the MRI, how your work is different from the MRI diagnostic.
Yeah.
So usually the MRI or the different type of tests, they are happen to be conducted on the later stage of the disease. So what we are trying to suggest is like we are trying to build an app based on the spontaneous speech and the linguistic characteristics, so that it can be there in the mobile app of a user, and it is continuously monitoring the cognitive decline. So when there is an onset of that decline, it gives you the indicator.
Okay.
So the MRI is
The last
later one.
later
Yeah.
one. That is
So the spontaneous, the onset is can be detected on the earlier stage. So that is basically the application of a model is like the early detection of the Alzheimer.
Okay. So if a person just sitting in front of your applications or maybe your system, so he is speaking about up to your questions, just putting to that guy, or even so is recording just be there in your application, and you try to know about from your system that yes, that person has symptoms of the Alzheimer's or maybe something like that. Is it?
No, but the thing is that right now the state of the art is like the model training has been done
Okay.
on an interviews.
Okay.
So that is a clinical test.
Okay.
But now what we are trying to do, we are trying to continuously monitor the spontaneous speech.
Okay.
Not only the interviews. So like the discussion that we are doing, the person is sitting in the classroom, the person is talking with the friends in the cafe, we are trying to monitor them so that we are continuously discussing or interacting with the cognitive.
And just to add up what Omar has actually explained so far, what we have done is we have already identified somehow the disease susceptible individual to Alzheimer based on our control data set. So you can easily see some physiological changes in your brain through changes in MRI. And what we are planning to do is just to pinpoint those individuals and have them like tested through our tools that we are going to develop based on acoustic or digital marker detection. So if you being a control, having some MRI based images that are showing that there is some physiological changes, so you can test yourself through the acoustic whether am I gonna actually have the Alzheimer later age, and our genetics is strengthening this up by providing the disease susceptible loci association with the MRI findings as well as acoustic digital markers.
Excuse me, I have a question. How it will deal with distortions? Like in real life, there are a lot of distortion. Like someone is talking and it's a lot of distortion. It can classify wrong. It can give wrong prediction. How it will deal with that?
So that's the noise basically. So we have talked about like we are trying to read it's just the audio set. So they are trying to just catch
They
the
will be taken care of in the pre-processing stage.
So background noise is everywhere. Any experiment you have performed with the background noise.
They can't even detect proper words. They can't even detect proper words. There are a lot of discursive market. How it will deal with that distortion? Sometimes we are driving car. It can detect. It can give wrong predictions. Like it can give Alzheimer. Simple normal human as an Alzheimer patient. So how it will deal with?
so basically there is a differentiate in hardware as well. So you need to select a particular hardware that can be only audible when the patient or the individual is near. So that's the one way of canceling the noise.
And one thing that was actually skipped is we have developed the hardware also for acoustic detection.
You can. The first one is decided edge here, like it's deployed on Raspberry Pi. For that particular part, can you please explain like how it was done? Since it's random forest and there we have to set the number of taps for the number of trees. And if the complexity or number of trees, so how is it being catered here? How will you deal with it? Or was there any, is it only for the inference part or did you guys deal with the parameters of the model? And did it impact the accuracy or the output compared to the server-side or cloud-side modeling? And the second one is for audio understanding. If you have you guys used any base model or you just train a classifier or you use models like Hubert or Wave? That was the first.
Okay, so the first part of the question is like we are trying to extract the relevant features, trying to make the model lightweight in that sense. And the second one is like we are trying to compare with the cloud and the edge as well. So this times that we were seeing it, the graph that we have shown, it was only related to the inference time, like on the testing. So on the training side, we have GPUs that can be used, so that does not matter a lot when you are in the field trial. So basically, what we have tried to develop a lightweight model in terms of the inference that is on the testing side. So in that way, we have used like a Raspberry Pi that is having a very low reconstraint devices. And for that, we have used like software optimization techniques like LoRa quantization. So that is what we have done so far. But in the next one, what we are trying to do, we are trying to evaluate them with respect to multi-modalities, because this is just like a single modality that is on the spontaneous speech. So when the genetic data is coming, when the MRI data is coming, so you need to make the model more robust. So that in that case, maybe we are trying to evaluate those technologies that you have just mentioned.
And one more, like, extending to this question, there is a parameter about the loudness in the acoustic and the audio. So, what if I speak with my mic closer to my mouth and there are scenarios where the mic might be
far away from me. So in that particular sense, the waveforms will be different for the loudness. So how will this be catered, or is it an edge case, or how will it be catered?
So in that case, because we do not have just one feature of loudness, we are having intensity, spectral, linguistics as well. So if you are saying that the mic is very near and the loudness, having a very good score, so there are another features as well that is differentiable. For example, number of pauses, the number of speech rate, and we are having spectral features, and we are having the frequency related features. So it is not only trying to predict the patient only on a single variable or the single feature. So if one of the feature is having a very good value, does not mean that he is going to
And for multiple features.
predict.
Okay, guys, there is a short break, tea break. You can discuss over the cup of tea, and you will come back here around 11 o'clock. They will continue this session. सर आपकी नजर आपको
Yeah.